right, so just to recap for you guys, when we left off on Tuesday, we were looking at some basic cell structure. We spent a little more time about the cell membrane and just saying it's a membrane, right? We said we looked at detail of some different types of proteins that you might become familiar with. We began to differentiate an integral versus a peripheral protein. We mentioned cholesterol and etc. So after using that visual introduction, I want to go through and complete the rest of your guided notes and add some text detail to that image. And just to give you a heads up, we're going to talk about some proteins coming up, and there's a table that spans page 2 and 3, and you'll see an image. So the next image that you see about proteins in a membrane are on page 3 of your guided notes. If you want to flip ahead, you can do so. But we use that kind of black and white image to look at some different structures in the membrane. And now we're going to add some more detail. The first thing that we're going to review is the structure of our phospholipids. The image on the left is giving a bit more molecular structure to the phospholipid. And the image to the right, kind of the generic image you've probably seen a lot leading up to now. A phospholipid, how did we describe a generic phospholipid? What term can we use to talk about the solubility of a phospholipid? Did we give? I may not have given it. The what? Amphipathic. Did we use that on Tuesday? Thank you. All right. So amphipathic is a term that we can use to describe the solubility about phospholipids and other like molecules. What does that mean? Amphipathic. Very good. It means that this molecule has both, or each, a hydrophilic region and a hydrophobic region. And if you recall from Tuesday, we talked about the heads are this and the tails are that. That's what we sort of taking all that time. We could have just said the molecule is amphipathic. The head is what? The head is hydrophilic, water-loving, water-soluble, charged. And the tails are hydrophobic. Those are where your fats are. So those are the water repulsive type components of the phospholipid. So a given phospholipid molecule is amphipathic, which is good that it's partially hydrophilic and partially hydrophobic, contributes to the function of that thing. Part of it has to interact where there's fluid, and part of it has to create a barrier. And that's what it does. And you'll find it arranged in what's called a bilayer. You'll find all these molecules arranged in a bilayer, 2 layers of them. So the bulk of a membrane is an assembly of phospholipids arranged in a bilayer. But a cell membrane is not exclusively made up of phospholipids. Dispersed throughout, you're going to also find various types of proteins. And we know that proteins, we said generically, may be integral or peripheral or have carbohydrate groups or something like that. But at this point, we hopefully understand that there are proteins in the cell membrane to do some job. Proteins are there to do some type of work. And that's what we're going to do, is summarize what types of work a given protein, when associated with the cell membrane, what it might be doing. Okay, so this list that we're going to go through, just like any other list we'll hit this semester, is not all inclusive. It's just kind of a snapshot or a survey of what's going on. So in this image, we can see some nerve cell membrane, and on the bottom left hand is a key for the different types of proteins that you might find, not only in the cell membrane, but all of our membranous organelles inside of the cell are going to have proteins, like a Golgi apparatus is going to have proteins in it. The refi, they're all going to have proteins associated with their membranes. The type and number of proteins is variable in the organelle or the cell type. And so my abbreviation here, M, stands for membrane proteins. This is just a generic image, something that I put together, so it's not real super artistic, but kind of getting the idea of a phospholipid bilayer. And then we can see plenty of integral membrane proteins and as well as single peripheral proteins. So let's review the peripheral protein. How did we define a peripheral protein? It's marginal, so it's at the margins of either side of the membrane. Good.
Loosely bound to the integral proteins and phospholipids?
Loosely, very loosely. And I'm even going to modify your word, I'm going to say attracted. Bound is okay, right? But I don't want to imply like cinching or real connectedness. It's more of an electrical attractedness than anything, to get technical. Okay? And so the loose would be the key part. They're loosely associated, but they're still at the membrane. What are they attracted to? It's an electrical attraction that they have. And so the electricity sometimes we can transfer or replace the word attraction with the word bound, but I don't want you to confuse like a covalent bond or something like that. It's not an actual covalent bonding, it's an electrical attraction that's being there. So that implies then the loose connectedness. Okay, so it's there, it's just not embedded in the membrane or anchored or anything like that. And so what color would my peripheral protein be in this image if you have color sided? The purple thing. I could have just as well drawn it in the extracellular fluid. And so a primary category of our peripheral proteins, you can call generally some enzymes. It may serve as an attachment, but a lot of these are enzymes. What might an enzyme do? It may break down, they may synthesize. That's, if you were to keep it generic, you would say they're a catalyst. They speed up reactions, whether they be breaking down or catabolic or creating anabolic type reactions. So they're just there for chemical reactions. In this case, that would be happening at the membrane. That's just one example of what a peripheral membrane protein may be doing. Most of our membrane proteins that we're going to see are actually integrated or integral into the membrane. So, actually, let me back up. Our peripheral membrane proteins, how might their solubility be? Hydrophilic. Make a note that your peripheral proteins are going to be exclusively hydrophilic. On the outside of them, they're hydrophilic. And so then we could take away, if there's no hydrophobic region, there's no way that they could actually integrate into the membrane. So they're peripheral proteins. Along those lines, then, how could we chemically classify an integral membrane protein?
Embedded in phospholipid layer.
Wait, it is embedded, but chemically, it's going to be, we're pointing at the new word, amphipathic. Your integral membrane proteins are amphipathic. And what that means is what? What does that translate to, Wes? What were you saying?
Polar and non-polar.
Yeah, polar, non-polar, and at the membrane it allows them to do what? What'd you say?
I erased it.
Oh, don't erase it. I was just asking a question.
That was wrong.
You were right. Just weren't there yet. If a membrane protein is amphipathic and is polar or non-polar, what does that mean to us? It can penetrate that membrane and embed in the membrane.
Embedded in the phospholipid bilayer.
Because our integral membrane proteins exhibit amphipathic nature, they can embed in the membrane. They have a hydrophobic region near the tails of the membrane, in the hydrophilic region facing either the extracellular fluid or intracellular fluid. You may recall from our introduction on Tuesday that our integral membrane proteins don't have to span both layers. Remember that? We found that they may just be on one we call leaf or layer or the other. And so they could be more simplistic than what's shown here. So I could just say draw protein here embedded in half of the membrane. Does that make sense? So our integral membrane proteins, because they're amphipathic, they can be integrated into the membrane. How much or how little depends on the protein and what it's designed to do. And so I have a snapshot of some things that proteins, what they might do when associated with a membrane. One function that an integral membrane protein may have is to act as a channel. What do you think a channel protein does? Very good. It allows things to enter and exit the cell through its little channel.
So the things that will be like entering or exiting through the channel protein, do they also have to be amphipathic, or does it?
That's a great question. The question is, well, what would be moving in and out of a cell through said channel protein? Does it have to be amphipathic, or what's going on? What type of solubility do you think, what type of solubility particle might a particle, have that would be moving through a channel?
Can be specific to the protein channel that you're talking about.
It'll be specific.
But some do allow just totally different things to come through.
Usually probably something that's hydrophilic or polar. So something that's polar, like water, like sodium ion, right, all your ions really, can move into or out of a cell through a channel. Because otherwise, a polar particle might be able to interact with the head, but what's going to happen as soon as it tries to get through the tails? It won't die, it's not alive, but it's going to repel out, right? Electrically, it's going to be shoved out. So normally sodium ions or fill in the blank ion or water are just not going to have an ability to naturally diffuse across the membrane. And so we need ions, we need water to get in and out of a cell. And so our cells have what? Channels. And they are specific, as Levi addressed. So if I want sodium in, I need a sodium channel. If I want calcium in, I need a calcium channel. If I want water, I need a water channel that's called an aquaporin. Okay, so they're specific. Great. I put it there now. Told her to ask that.
So water has a specific channel as well as using the channel ones, the open ones?
Yeah. The channels that are designed for water are always open. They're called aquaporins.
Okay.
And so they're specific for water and they're always open.
Oh, so, okay, so there's different of the channels.
There are, as far as going, it's like I planted these questions. You guys are good. Where I was going next is the channels aren't always open. Sometimes, as you can kind of see in that image, sometimes they are closed. They're closed, though. They could probably be open. So let's differentiate those. Channels that are always open, okay, they don't have the ability themselves to close. We'll refer to those as leak, or they call them leak channels. Conversely, other channels can be regulated, closed or open. And what, does anybody know what those are referred to as? The channels that we could regulate? They're called gated channels. Some channels are always open. Those are called leak channels. And some channels are gated, so they open and close. A natural progressive question is, well, why would a channel ever have a gate on it? Why? Why do you think we need a gate on some channels? Regulation. We need to regulate what enters and exits on certain specific molecules. So those gates there are to regulate the entry or exit of whatever is designed. These two types of channels are going to be the foundation of really understanding the nervous system and the muscular system. So about 50% of the class is depending of the class material is dependent upon understanding a leaky channel and a gated channel. And then we'll look at specific kinds, gated sodium channels, right, and we'll just keep on. We'll become introduced to those when we first study the nervous system in lab coming up in a couple of weeks. What color would be of a protein here might be a gated channel to be represented by? Kind of the red thing and the green. These channels are actually what at this point? Closed. So even though it's designed for, say, sodium at that moment in time could not diffuse, facilitate through that channel. Only when something opens could a particle facilitate through.
So what's the difference between like your blue and yellow representation?
Nothing. I just pulled this from one of your handouts that come up. For today, nothing. When we do our our handouts, one is for sodium and the other is for potassium. And we'll learn that they move in different directions. And so it'd be specific. So this is sodium, potassium. I think sodium, potassium is how I'll illustrate them in the handouts. But at today, there's no difference besides a leak versus a gated. Some integral membrane proteins may act as carriers, which sounds a lot like a channel, but there's a big functional difference between. What do you think a carrier protein might do? It'll actually physically carry it, and what that implies, then there's got to be a bond. Okay, so a temporary bond and a transport. And so a carrier, as the name implies, carries a specific molecule across a membrane. It's a transporter as well. And, channels technically are transporters too. They're to carry a specific molecule. A carrier we're going to study, we'll talk about are called glucose carriers. They carry glucose into a cell. And then in discussion in class yesterday, someone said, well, don't they have to use ATP? Well, that depends. Depends on what it's designed to move and how it works. Okay, so when we get into chapter 4, we're going to learn that some carriers don't need an input of energy. So that would just be another type of facilitated diffusion, while some do. And that'd be a form of active transport. So we'll differentiate whether or not ATP is needed when we get into chapter 4. But right now, we're just saying a carrier carries something, a specific something, across a membrane, because it's a specific integral membrane protein. What type of particles do you think might need to be carried? Hydro. Well, if something's hydrophobic, really it could just simply diffuse. So, hydrophilic. Yes, well then 50-50, yes. Hydrophilic. And things that are maybe just, I heard it. I thought I heard it. Things that are hydrophilic or maybe just very what? Large. Like glucose. To address the hydrophobicity, something that's hydrophobic is probably not going to have any difficulties, what we call dissolving or diffusing through that hydrophobic region of the cell. So generally wouldn't need a transporter for them. But bigger particles or polar particles would meet a carrier. Another foundation important for understanding, comprehending, I guess, what's going on this semester is the role of receptors. Receptors, as their name implies, do what? Receive. So some of our integral membrane proteins may serve as receptors. Receptor is almost as generic a word as protein. So receptor protein structure is very variable or diverse as well. They may be very simple, they may be very complex. They may be part of a channel. The receptor may be on the channel. To regulate that channel's what? Gate. So a receptor receives a specific molecule. How it works just depends on how it's built. Just going along with our examples, we talked about insulin, right? Insulin, for your bodies to listen to it, your cells need insulin receptors. If you want your skeletal muscles to listen to the neurotransmitter acetylcholine, they need acetylcholine receptors. You want your brain cells to listen to dopamine, your cells need dopamine receptors. So some of our receptors are integral membrane proteins. Please note that not all receptors are membrane bound. We're just looking at the membrane right now, right? There are other receptors inside of the cell that it would have a different function. Some of our membrane proteins can serve as points of attachment for something called the extracellular matrix, which I don't have represented here, but as the term implies, have you used the word matrix before? So, other than the movie, you use the word matrix. Like a grid, right? And that's a good visual analogy. It's a grid of proteins where? Outside of the cell. So extracellular matrix is a bunch of proteins right at the margins of the cell. They attach to our membrane proteins, and they're there to give physical structure and other types of things. Where you're most familiar, have you heard of, say, collagen? That's very common for extracellular matrix proteins that the average person knows or has heard of, right? We hear about it in beauty products, right? This getting rid of wrinkles and etc. So because this extracellular matrix is there, it's scaffold well in the trap water and cells look cool and all kinds of good stuff. So that'd be an example of a protein. The extracellular matrix attaches to our membrane. So not all cells have a well-developed extracellular matrix. You find it most prominent in like your connective tissues, for example. Additionally, we have some membrane proteins that serve in the role of what's called cell adhesion molecules. This one too kind of gives itself away if you kind of pick apart at the nomenclature. So these are molecules that allow for temporary bonding, binding, adhesion, sticking of one cell to another. You may have never used the word cell adhesion molecule before, but you've appreciated them. Have you ever heard of your macrophages? What are they? What is a macrophage? It's a type of what?
Eater.
What does it eat? What is
Bad
it?
stuff.
Foreign bodies.
Yeah.
Pathogen. There you go. Pathogen sounds good, better than an eater. The buffet is like. It's more than that, right? It's not just a nutrient type of thing, but it's part of which system? The immune system. And it goes around and it's kind of like a watchman. It rolls around and checks around, looks for foreign particles. The way it rolls around is using cell adhesion molecules. It temporarily touches proteins to other cell adhesion molecules. And it's almost kind of, it's almost like a rolling handshake. So if you guys were cells and you had your cell adhesion, go ahead and stick your cell adhesion molecules out. I could roll around and I could grab each, if I were the macrophage, I could use my cell adhesion molecules and I could roll around the entire class, right? Your cells don't have arms, they have the adhesion molecules, which are just specific types of proteins. So they can use temporary attraction, they could use more long-term interlacings with the extracellular matrix. They may serve as receptors, carriers, channels. We keep going on. Some proteins in a membrane are still enzymatic, so some of our integral membrane proteins are enzymes as well, having the same capacity, carrying forward a specific reaction.
Is that cell adhesion, is that like cilia?
It is not like cilia. Cilia are membrane extensions, so there's a protein, but there's still membrane encompassing it. The cell adhesion molecule would be an integral membrane protein, so the membrane doesn't surround it. So it really is, it's almost like the track on a track tire, like a bulldozer or something like that. Does that make sense? So the tires are always, the ridge part's kind of sticking out. So, I don't know if that was a good visual or not. I was like, what is she talking about? But it's not, the difference between a cilia, well, there's many structural differences, but the big difference is a cilia has the membrane around it. So it's a protein pushing through, like a hand through a glove. In this case, adhesion molecule, there would be no glove, just a hand sticking out, so the protein exposed. And it's not for movement of fluid, it's just for like an anchor on a boat or something. Not moving the boat. Not moving the cell. Another important component of a cell membrane, besides the phospholipids, besides the proteins, is a molecule called cholesterol. And you know, cholesterol gets kind of a bad rap out in society. Because we're human and we tend to overconsume things. And so cholesterol gets a bad rap because we overconsume it and it causes heart disease and etc. However, it's not something that you would ever want to take away from your diet, right, 100%. Because it's important for membrane structure. We can make some of it, but it's okay to take it in moderate amounts, just like everything else that we consume. So cholesterol, shown here by the two little golden four-ring structures. How about we describe its chemical solubility or water solubility? It's mostly hydrophilic, but there's a little bit of that amphipathic. Somebody yesterday was calling it pathetic. It's not pathetic. Amphipathic. Cholesterol is amphipathic. The bulk share of it is hydrophobic, but there's a tiny bit of it that's charged polar. So it is an amphipathic molecule. The question is then, well, why is it there? Somebody know why it's there? To add elasticity. It kind of, I kind of like that word elasticity a little bit. It has something to do with temperature, though, right?
Also, like whenever it's colder, it'll help maintain fluidity, and when it's hotter, it's going to help maintain rigidity.
Very good. So what he said is cholesterol. I'm going to simplify it and then repeat what he said. It's basically like a temperature buffer. It allows your cells to be exposed to a little bit wider range temperature. In cooler environments, it prevents your cells from gelling. In warmer environments, it prevents the cells from kind of washing away. So it maintains the fluidity of the membrane. It maintains the fluidity of the membrane and widens the temperature range that a particular cell might be exposed to. So
does it help keep the membrane fluid?
It helps keep the membrane fluid. Very good. Those are the three major components of a cell membrane. And we talk about phospholipids, proteins, and cholesterol. Now if we go look at any other cell in your body, we need to kind of see what else there's about a cell. And with the rest of the cell, internalized, you can refer to as the cytoplasm. The cytoplasm is kind of a generic word synonymous to our intracellular fluid, right? The internal environment. So it's the mixture of everything inside of the cell. So it's the contents of the cell. Cytoplasm is just a generic term for what's in there. It includes three primary components. The three major components of your cytoplasm include, one, your organelles. So your cytoplasm, the first major component of your cytoplasm are the organelles, which we'll go through and address coming up in a second. Additionally, the second major component of your cytoplasm is called the cytosol, S-O-L, C-Y-T-O-S-O-L, cytosol. That sounds confusing because the prefix cyto is there. But cyto just means cell. Okay? So plasm, the fluid, sol, the solution. So the cytosol is the liquid, the actual just liquid part of the cell. So exclusively just the liquid part. So the water and the solutes that you might find suspended in there, the ions and whatever else is in that particular cell. So the cytoplasm, three major components. The first is the organelles, we'll address. Secondarily, the cytosol, which is the solution. In other cells, you may find also a third component, just generically referred to as inclusions. Inclusions is another type of general area that you might find in a cell. Inclusions. I hope that kept going. And some examples of inclusions that you'll find in a cell would be like our pigmentation. What do we call that? What's our pigmentation referred? What's the name? Melanin contributes to our skin color. So some people have less, okay, inclusion, and some people have more inclusion. Okay? And so we could say melanin, or we could say melanin inclusion. So your skin pigmentation would be an example of an inclusion. The glycogen stored in your liver cells or your muscle cells are examples of inclusions. The fat stored in adipocytes or fat cells are generically called inclusions. So the type of inclusion depends upon the cell type. So the cytoplasm is comprised of not only our organelles or cytoplasm, but also various types of inclusions. What I want to do now is go on and look at the different organelles. I'm going to list them here. You don't have to scribble them down, but I do want you to go ahead and give a definition. We're going to go with the definition of those structures inside of a cell that do have a specific function. The qualifier that's going to make our definition different is that they have a membrane around them. So we're going to look at membrane-bound structures inside of the cell that have a specific function. This definition, again, someone sitting next to you may say, well, that membrane addition to that definition really limits you, and it does, and that's why I like it. But they're less short to study these things. But what that tells us is we're going to have to study non-membranous structures inside of the cell and what they do as well. So let's first start and look at our membranous organelles. We're going to look at the nucleus, the smooth and rough ER, Golgi apparatus, lysosomes, peroxisomes, and then conclude with mitochondrial structure. The first organelle that we're going to study is called a nucleus. You've heard of it. Maybe you've not really understood the structure of it, but hopefully you at least have an idea about what it does. It has something to do with what? Genetic information, genetic material, genes, right? Our genome is contained inside of the nuclei of each of your cells. The nucleus is critical to cell function. Without a nucleus, the cell wouldn't be able to divide, and it wouldn't be able to make proteins. We know that cells need proteins. Those codes come from here at the nucleus. In this image, this is a nucleus, and the rough ER kind of pulled out. So this image is not an entire cell. Okay, so it's just a little nucleus kind of picked out. And what I want to do is look at the nuclear structure in a little bit better detail. The first thing to note about the nucleus, not only is it membrane bound, it has two membranes about it. It's double membraned. The nucleus has two membranes surrounding it. The phospholipid bilayer, right, with proteins and right, all that is here. It is wrapped around. There's two of them. So we're probably protecting something pretty important, right? Yes. All right, so not only is the nuclear envelope double membrane, connecting the two membranes to create kind of like channels are these things called pores. These golden structures right here are called pores. Going along with our thought processes earlier, they're kind of analogous to like a channel. So what do? They regulate and allow specific molecules to enter and exit. We'll just call nuclear pores. Right, those of you in cell biology will look at the structure. It's very complex. It's a bunch of proteins assembled together to form each nuclear pore. All right, so our nuclear envelope's double membrane. They have pores. They also have what on them? Ribosomes. Ribosomes, which we'll study coming up, if not today, definitely on Tuesday. But what do the ribosomes do? Something to do with protein, synthesis of protein. So we have ribosomes attached to the nuclear envelope that help assemble proteins. Translation. They had a question. What else? All right, so the nuclear envelope, double membrane, pores, ribosomes. Those are the major features about a nucleus. The envelope structure. Inside of the nucleus, we said I would have our genome in here. When the cell's not dividing, the genetic material is usually arranged in what we call chromatin. So it's kind of partially packed, but it's not condensed into a chromosome. In addition to the chromatin, there's generally one or two clusters that are easier to see with a transmission electron microscope, a dark staining area inside of the nucleus, which is called the nucleolus. The nucleolus, sometimes if you're not familiar or you're just kind of learning this, it almost looks like that might be the nucleus and the rest would be the cell. But here's the thing, it's not. But two, the nucleolus is not membrane bound. It's just a concentration of molecules. Does anybody know what's going on in the nucleolus? What these molecules are doing?
Creating proteins.
Our ribosomal RNA synthesis to help pre-manufacture our, not the chromosomes, the ribosomes, so that we can make our, so it can go on and make our protein. If you recall, we mentioned on our first day that cells can't go out and shop for what they need, right? And if we know that ribosomes make proteins, but ribosomes have to get made somewhere. So since they're part protein, they've got to get coded for and pre-assembled in in the nucleolus region. So we're pre-assembling our ribosomes.
You said it was like a collection of
I just said molecules,
molecules.
whose job is to pre-assemble our ribosomes. Full assembly would happen out in the cytoplasm.
And what did you say about the chromatin?
The chromatin is the DNA, loosely packaged.
Shake and bake. DNA, that's what's up.
I don't know. Your DNA can exist in different states, like fully condensed in a chromosome when you get ready to divide, or fully decondensed if you're like transcribing something. But it makes so much genetic material in the given nucleus. It's just like the clothes that you have in your house. It's easier. Sure, you could lay them out all in sequence, right? But it would take up your whole house. And so it's best to partially package them in, say, your dresser. So you don't have to put all of your clothes in one dresser drawer, but you could put it throughout, right? So it's not super condensed, but it is kind of it's organized. And so it's the more organized, intermediate organized structure of your chromosomes. All right, so the nucleus, right? It's got a really cool structure. And now I want to go ahead and move on to the smooth ER. You can abbreviate it ER if you're okay with doing so. And the term smooth doesn't have to deal with its function, but its structure. So under an electron microscope, a TEM, it visually looks smooth. Okay, so it looks not rough in comparison to usually its neighbor. The smooth ER, it may be continuous with the nuclear envelope or it may be offset. It just depends on the cell type. It may be more simplistic. It may be very well developed. It's always described as being tubular. So if you can look back here in the back of the image, you can see the tubes. Not forgetting that those are phospholipid bilayers with proteins, right? And on the inside of each of these organelles, something is going on, something specific is going on, and that's what we're focusing on. The smooth ER that we're going to, the function of the smooth ER that we're going to study, focus on, is the role of the smooth ER in calcium storage and release. The smooth ER stores and releases calcium. So what is calcium? In the body is what? We do need to intake it, for sure. We don't really use it for directly energy conversion though, like you might take it in lipids. So, structure for things like bones. What is it though? It's a mineral. And it's carried in the body in what form? An ionic form. It's an ion. And we want to store and release it across a membrane. How do you think it gets across the membrane of the smooth ER? Good. All right. Very good. So because the smooth ER has calcium channels, it can store and release it. So please note that the smooth ER has calcium channels. To allow for the calcium to move from the lumen of the smooth ER to where? Where might calcium go? If it's in the lumen of the smooth ER, where might it go? To where? I heard, but I didn't understand. Where might it go? If it's in the lumen of the smooth ER, it goes through a channel, where would it be next? The rough air and the smooth ER aren't physically connected, so what happens is it's just the fluid that's next to it is the cytosol. So calcium ions will flow from the inside of the smooth ER to the outside, to the inside to the outside. We'll put it to use in the muscles. So because the smooth ER has calcium channels, we can regulate calcium flux between the smooth ER in the cytoplasm. You've probably studied the smooth ER more in its role of lipid metabolism, so our steroid, right, free biosynthesis, and etc. It definitely happens here in the smooth ER. And the precursors of our peroxisomes, also assembled here, in the smooth ER. Any questions about the smooth ER before we move on to the rough ER? If we contrast that to the rough ER, the term endoplasmic reticulum is dealing with just the network of membranes. So some are smooth and some are rough. We described as a smooth ER as being two. But if we contrast that to the rough ER is more, how would you describe this? It's kind of like rows or walls. They're studded, right?
I said folds.
Folded, right? And so the terms that's usually described, like you'll find in your textbook, flattened sacs, more flat than tubes. And what's on them, giving them a rough appearance, are ribosomes. And so under a transmission electron microscope, the rough ER looks rough. It's got all these little ribosomes fixed to it. What does a rough ER do? What's its job?
Binding site.
Binding site for the ribosomes. And because there's a binding site there, what happens? Proteins are made. So translation happens here. Oh, sorry. Directionality, though, is very, very important to understand. At the rough ER, when proteins are made at the rough ER, the peptide grows and extends into the rough ER lumen, not out in the cytoplasm. So when a protein is made here at the surface of the rough ER, when it starts assembly here, it's actually going to grow and extend into the lumen of the rough ER. So when you put it that way, it almost seems questionable. Why would you make a protein and just keep it trapped inside of an organelle? Here's the thing, it doesn't stay trapped in the rough ER. It just gets assembled there. And once it's assembled, can you see right here? It's called a cistern. The membrane of the rough ER buds. We use the word bud, okay? The membrane of the rough ER buds off, and the newly synthesized proteins are now inside of that, what we'll call vesicle. So the rough ER manufactures some proteins, and then entraps them, if you will, packages them in a vesicle. Vesicle. And forms vesicles, which are just little pieces of membrane. In this case, we have proteins inside of them. Fair enough? We got a protein wrapped in a vesicle. What can we do with the vesicle now? We can move it. And that's exactly what happens. Most of these are going to move to the next organelle that we're going to study called the Golgi apparatus. I've got a video coming up, so it might be useful to just kind of turn your attention here. It's like a 16-second video animation, so it's super fast. I'll replay it. But I want you to notice this is showing the Golgi body or the Golgi apparatus. And notice a vesicle is going to be transported from the rough ER to the Golgi. The proteins, which would have originated the rough ER, are shown in the blue, yellow, green dots. Okay, and so I want you to notice the migration. Right, so we have a single Golgi apparatus. Coming from the rough ER are little vesicles with proteins in them. Notice how the vesicle fused with the Golgi. The proteins are kind of processed through the Golgi and then repackaged into new what? Vesicles. Let me show it one more time. Vesicles containing newly synthesized proteins at the rough ER are transported over to the Golgi apparatus. where we're going to see they'll be modified and processed and then repackaged into new vesicles. Does that visual help a little bit about what happens? It's not as random or accidental as it looks like, or magical, like those vesicles like, woo, Golgi next, right? It's very much regulated. There are protein tags to signal where that vesicle needs to be shipped. It calls into action the microtubules that you kind of studied in your handout. It's very much regulated. But the concept is true. Where that vesicle goes to the Golgi and it dumps its contents into the Golgi. The question is why? What's going on with the Golgi apparatus? What happens? They are at the very end. They're packaged, but why are we pushing them through the Golgi?
For the translation process.
To change them to what?
Process.
To process them, maybe what else? Modify them.
Tag them for the destination.
Tag them, right, for where they need to go. So all kinds of modifications may be happening to a protein when shipped over to the Golgi apparatus. So what that tells us is when they come from the rough ER, they may not be fully ready to go. So we can make some additional modifications in the Golgi apparatus, and that's what it does. Kind of like a quality control, but there's actual still determined. It's not like a proofreading kind of thing. It's more of a next step, next phase of manufacturing. So, secondary processing maybe is another way. I don't know, so I don't know if the statement is true or not. I don't know if there's any quality check going on in the Golgi. I believe the quality check would happen at the rough ER. But there sure could be, but I don't know. But it's more of a, remember we talked about glycoproteins last time? Those big carbohydrate groups are added initially at the rough ER, but they're big blank trees, if you will, carbohydrate trees, and so they get cooked and modified for the right structure there, and those kinds of things happen. So the Golgi apparatus modifies proteins that have been manufactured at the rough ER. I want to step back now that we have this relationship between the rough ER and the Golgi. I want to step back and look at the rough ER and add a little bit more detail. These proteins made at the rough ER, modified at the Golgi, are going to end up at some point in that cell's life in one of two locations. The fate of proteins made at the rough ER is going to be either, one, integrated into a membrane to act as a channel, transporter, receptor, or whatever. They're either going to be integrated into a membrane if they are what? It's a protein. Amphiphilic. If the protein which was just manufactured is amphiphilic, it's going to be integrated into a membrane. If it's hydrophilic, it's going to be secreted out of the cell. For example, like a hormone. Hormones, to get out of the cell, if they're proteins, need to get packaged into a vesicle to kind of purge them out into the extracellular environment. So what this tells us is the rough ER doesn't make all the proteins for the cell, only those that are going to be incorporated into its membrane, or those are to be secreted from the cell, like a messenger, hormone, neurotransmitter, or something like that. Proteins that have a job inside of the cell, like your cytoskeleton that you studied, or maybe some cytoplasmic enzymes or something like that, get made in a different location. We'll clarify that. Looks like, if not today, definitely on Tuesday when we come back. All right, so the rough ER makes proteins, yes, but does it make all the cells proteins? No, it does not. It can help also initiate the manufacturing of our lysosomes, which is another type of organelle. It's a membrane bound. It almost looks like a vesicle, like we've described before. And that's pretty much what it is. However, we do know the type of proteins that are in there. What is in our lysosomes? Enzymes. You know what type of enzymes? Hydrolytic, hydrolases. There's a family of enzymes called hydrolases. And hydrolytic is what they do. What does a lytic mean? To rupture, to break. They break chemical bonds. They break down substances. Our lysosomes have enzymes in them called hydrolases. And they're hydrolytic. What that means is they break down substances that maybe are excessive or potentially damaging. Classic example of the lysosome is again in the macrophage, which we talked about previously, the white blood cell that kind of scrolls around and looks for pathogens. If it takes in a foreign particle, it can fuse that foreign particle with the vesicle. So when those membranes fuse, all these hydrolytic enzymes digest and break down or lyse whatever it was. Whether it was good or bad or whatever, it's breaking down. If it's exposed to those, all of those hydrolytic enzymes in there. It's like a little bit of, sorry. A similar type of organelle is called a peroxisome. And our peroxisomes, same type of shape, they're kind of spherical looking vesicles. They also have enzymes in them. They have two classes of enzymes. Okay, so same kind of concept. What are they going to do? Break down, degrade, okay? Substances that are potentially toxic like alcohols. There are two types of enzymes that you would find in a peroxisome, two families of proteins. They're actually called families. Oxidases and catalases. Anytime you see the suffix ASE, that tells you that's an enzyme. And some enzymes do what? Carry out what type of reaction? Oxidation, right? Very good. And some catalase, okay? Catabolize, break down. So what they do is alternatively, one makes H2O2. What is that? Hydrogen peroxide. So when exposed to a toxin, catalases begin to work on the molecule, break it down, and in doing so, it releases hydrogen peroxide. Have you worked with hydrogen peroxide before at your home? What happens?
Bubbles.
Bubbles. Very reactive, right? So we really don't want to have all of that going on inside of a cell, right? And so what happens is, as soon as that hydrogen peroxide is made, catalases do what to the hydrogen peroxide? Split it apart. Into what? H2O2 could be broken into water and oxygen. So they alternatively make, then break hydrogen peroxide.
Catalases make and break both?
No, oxidases exclusively produce the hydrogen peroxide. The oxidases make the hydrogen peroxide. And the catalases break it, split it into what? Water and oxygen. Yes?
Back to the lysosome, I have a question about the hydrolytic. Is it hydrolytic because if it breaks it down, it pulls the water from it and then?
Water is liberated.
Okay.
Yes. And the same thing here too with the catalases. But they exclusively use the water. Yes.
You said water is liberated in hydrolytic? Ain't that using water to split it? So you're just like shoving water in the middle?
Hydrolases. We should be liberating water. I'll double check myself.
I thought hydrolytic is when you take the water, it's water splitting, you use the water to split it, and condensation.
I'll double check myself because I easily get confused when I'm talking. So I will go back and double check that. If it's used or made.
I thought I read that the water